Done, sir.
Okay. Good evening, one and all. I am Himani Tabale, and as a convener, I welcome you all on behalf of Microbiologists Society India. Today's session, we are going to focus on bioactive peptides from fermented plant-based food, current trends and challenges. Let me welcome our speaker for today's session, Dr. Subhrata Hathi sir, Assistant Professor, Department Dairy Microbiology, SMC College of Dairy Sciences, Kamdenu University, Anand, Gujarat, India. Dr. did master's and doctorate from National Dairy Research Institute, Karnal in Dairy Microbiology. He also did postdoc from the University of Queensland, Australia in 2018 under the Indo-Australia Career Boosting Gold Fellowship sponsored by DBT, New Delhi. He has published more than 130 research papers in reputed journals nationally and internationally, 10 review papers and 3 edited books. He is also the editorial board member of Scientific Reports, Frontiers in Nutrition, Journal of Dairy Research, Discover Food, and Indian Journal of Animal Health. Dr. Hathi sir is a recipient of numerous prestigious awards, which include University Topper as Gold Medalist in Undergraduate Program, Best Teacher Award 2018 by ICAR and AAU, Anand NAAS Young Scientist 2019 Award sponsored by NAS New Delhi, INSA Visiting Scientist Award 2018 by INS, and also the recipient of Dr. Gauri Ganguly Memorial Award 2019 by Indian Science Congress Association, Kolkata. Best Innovator Award by Microbiology Society of India, Maharashtra, and also visited the University of Copenhagen, Denmark, for presenting the research work under Indo-Danish Collaborative Workshop 2018. He is also the elected fellow member of National Academy of Science and associate fellow of NAS, WAST, and NATC. He also received BRICS Young Scientist 2021 by DST, New Delhi. He is also awarded with International IFST Award sponsored by ILS Europe on June 22. Dr. Hathi sir delivered an invited talk in Obihiro University, Hokkaido, Japan, as an international visiting academic. Recently, Dr. Hathi sir received the Best Teacher Award from Kamdenu University from the Honorable Governor of Gujarat during the convocation of the university. Without further ado, I request Brota Hathi sir to please proceed with today's session.
Am I audible?
Yes, sir, you are audible.
Okay, now I'm sharing the slide.
Your co-host Subrata sir, you can share the screen.
My screen is, I have to share, or it is one participant can share at a time that I have to click?
Sir, you can click on share. So,
Okay.
yes.
Now the PPT is visible?
No, sir, not yet.
It is not coming. Now
Sir,
visible?
there is, no, sir, not yet. Sir, there is an option of share.
Share, but it is not coming the share windows or share like these things.
Sir, when
Here,
you click on
when I click na share option, allow to share window or share screen I should do.
anything, you can share one screen or you can share the entire window, tabs, all of the tabs.
Okay. Now the visual.
No, sir.
First,
Yes,
you have
sir.
to open. Ah,
Now you've
okay.
started. Yes.
Visible.
Yes, sir. Yes.
Now it is in full screen.
Yes, sir.
Okay, good evening all, and I'm also thankful to Professor uh, Desmukh sir for giving me the opportunity to speak on a very important topic, bioactive peptides from fermented plant-based foods, current trends and challenges. And also, I am very much thankful to Dr. Himani for giving a very nice introductions. So, I will cover uh, about the bioactive peptides from its science to the applications by applying the plant-based materials, mainly plant-based foods, through fermentations, mainly the microbes. Maybe it's a bacterial fermentations, or maybe through mold fermentations. Okay, so depending on the foods, different microbes will be used to produce bioactive peptides, which are having a very good health impacts to manage different kinds of metabolic syndromes like hypertension, diabetes, then stress, mainly the oxidative stress, etc. We'll discuss one by one. So, to start with a brief introduction about the topic, plant-based foods known for their health benefits since long we know because we are totally surviving on the plant-based foods in a daily basis. Okay, because of their good nutritional values as well as they produce different kinds of phytochemicals and other uh, antimicrobial substances from the plant-based materials, which give us not only nutrition but also they improve the immunity of the human health and provides all kinds of minerals and vitamins. Everything we got it from the different plant-based materials. But here we'll talk about the fermentations, mainly the fermentation of plants material, plant-based foods through lactic acid bacteria. So all of you know that lactic acid bacteria have a GRAS status. They are basically safe and they have a lot of health benefits. We know about the probiotics. You know that most of the probiotics are from lactic acid bacteria, and these lactic acid bacteria are being used for fermentation of plant materials for making different kinds of foods. And when these bacteria will grow and they ferment the plant-based materials, these proteins of the plant-based material will get converted to the bioactive peptides, and these peptides will have a different health benefits to the host. So, as we are talking about the plant materials, but we should know that how much protein is there in different plant sources, because you know that peptides will be generated from the proteins. Peptides are nothing but more than the fractions of the proteins. So, what we eat on our day-to-day basis from legumes or cereals, tubers, pseudo cereals, nuts, oil seeds, they have a different level of proteins. If you talk about the cereals, then wheat flour, corn, barley, oats, rice, sorghum, and you can see that wheat flour has around 8 to 15% proteins. Then legumes, they are rich source of proteins like soybean, lupin, pea, beans, chickpea. Even in nuts also, you can see that almond has a around 21% protein. Then cashew nuts, hazelnut, peanut, they are rich source of proteins. Even oil seeds, some microalgae. So these are all plant based sources, and they have a good amount of proteins. And you know that every human beings they require protein for maintaining their health. As per RDA, how much your body weight, that much of protein in gram you require daily. So every human beings, they require proteins from plant sources or from animal sources, but we have to take proteins on a daily basis. So when we talk about the protein, but more important part of the protein is the essential amino acids. Okay, so all the plant materials, but in case of soybean, you see it is a good source of essential amino acids like lysine, tyrosine, isoleucines. Okay, it has a good amount of essential amino acids. Even corn, it has a very good amount of phenylalanine and leucines, even methionine. Then you can see rice. Okay, most of us we take rice on our lunch or daily, or on a dinner daily. So it is a good source of valine and methionine. Even lupin, it is a good source of histidine. So all the essential amino acids are present in a very good amount, okay, in milligram per gram of the total proteins, and they basically meet our daily requirements. Then what are the functions of these amino acids? These amino acids are essential for the synthesis of proteins, which are crucial for the structure, function, and regulation of the cells, tissues, and organs in the body. And some of the enzymes which basically break down the proteins and release these amino acids from the protein molecules, and these amino acids will be go into the blood circulation systems, and they will participate in our different active biochemical processes. So through metabolism, amino acids are involved in various metabolic pathways to produce energy or to provide the nutrients. And some of the amino acids are also used for the production of different hormones like insulin or other growth hormones. So our main topic is the bioactive peptides. So bioactive peptides are the protein fragments that benefit the body system and overall human health. Most of the bioactive peptides, their amino acid range in between 2 to 20, and their molecular mass is around 0.4 to 2 kilodalton. And how we can produce the bioactive peptide. This is a very important part in industrial level, that you have to produce. So you must have some production systems where you can produce the bioactive peptides. So bioactive peptides can be produced from the plant tissues or plant organs, but this method cannot produce certain peptides. Is special therapeutic. So you have to be very much, selectable to, particularly to choose a particular proteolytic enzymes for the breakdown or hydrolysis of the plant proteins. And then have you have to go through a particular protocol or a procedure through which you can generate the peptides which have some health benefits to the host. At present, the most commonly used methods for hydrolysis by digestive enzymes or the proteolytic enzymes are basically produced by the microbes or through or some plant sources also. So, bioactive peptides through fermentations. At present, we know the lactobacillus, lactococcus, or different types of yeast and even kefir grains. Kefir grains is the uh, full of microbes. Basically, it is the combination of lactic acid bacteria and yeast, and they're used for the fermentation of milk as well as the plant materials. And these lactic acid bacteria, they are very good they have a very good proteolytic systems, because the proteolytic system is the key requirement to use for fermenting the proteins and to have the bioactive peptides from these uh, plant proteins. And they have a very good systems in their cells by producing enzymes or by allowing these peptides to transport through different transporters embedded in the uh, cell membrane. And then these peptides, when it will reach into the inside the cell, then again it will be digested with the different enzymes, and finally they will release the amino acids. So the lactobacillus have a very good proteolytic systems, and it consists of three things. First is the CEP, cell envelope proteinase. This is the enzymes required to break down the large molecules of the proteins. Then peptide transporters are there. Basically, it is embedded in the cell membrane. And then there are some aminopeptidase or other enzymes, proteolytic enzymes present inside the cell to break down those smaller peptides to release the amino acids. So this figure will make you very clear about these, how these lactobacillus are basically important for producing the peptides from the proteins. So you can see here, this is a CEP, cell envelope proteinase. It is an enzyme, so it will break down this long chain of polymer of the amino acids to the smaller fragments. This is called oligopeptides. So these enzymes will break down this protein into the oligopeptides, and these oligopeptides will be transported by using this transporter, OPP, oligopeptide transporter. And when these oligopeptides will enter inside the cell, then other enzymes like peptidase or aminopeptidase, they will again break down into peptides or amino acids. And these amino acids will be utilized for process for production of energy or some metabolic purposes. So what are the advantages of microbial fermentation for bioactive peptides? This process is less costly than the enzymatic hydrolysis. Okay, because in enzyme you have to purify it, then the upstream and downstream processing is very costlier to purify particular enzymes. But here we will directly add these proteolytic bacteria. So when they will grow, they will produce these uh, different types of enzymes, proteolytic enzymes, and they will break down the proteins into the peptides. And another thing is that the organisms which we will basically use, mainly the microbes, okay, they should be also grass, having a grass status, because ultimately these peptides will for food formulations, for development of functional foods and as a nutraceutical. So we will have to for consumption. Any problems? Then
No, sir. No,
effect of
sir.
okay. Then effect of fermentation on various bioactivities of the plant-based food. So here you can see that this is the source of the different plant-based foods like wheat sourdough, then fava beans, barley, oats, okay, lupin, tomato seeds, and these are the different microbes are being used, maybe lactobacillus or rhizopas or pediococcus, or you can see here some bacillus. Okay, so these microbes are being used, and after fermentations they produce some bioactive peptides, and they are having a different biofunctional properties like antioxidative, antimicrobial, then anti-diabetic, okay, uh, anti-hypertensive, anti-proliferative. So these are the different biofunctional properties. You will get it from these bioactive peptides generated from these food materials after the fermentation with these very important microbes like lactobacillus fermentum or lactobacillus plantarum or pediococcus acidilactici or bacillus subtilis. Then health benefits, I already told that bioactive peptides have different health benefits. They are scavenge the free radical and give antioxidant activity. They also help in managing the blood pressures in our systems. I will discuss how it maintain or manage the blood pressure, what is the role of the peptides in anti-diabetic, anti-hypertensions. Okay, it also improve the digestive systems, and some of the bioactive peptides, they act against the tumor cells and prevent the cancer cells, means the conversion of normal cells to healthy cells to the cancerous cells. So these bioactive peptides have different roles, and people are working on it, and they are proving it. They're taking trials on preclinical or clinicals, and some of the peptide products are available in the market also. Then first I will discuss about the antioxidant peptides. So free radicals are chemical entities that are reactive and have one or more unpaired electrons. And free oxygen radicals play a vital role in our body's normal functionings. We know that when you smoke a cigarette or other things, the free radical generate in your systems, right? And that free radicals is very harmful because they attack the DNA, they attack your proteins, they attack the fatty acids, because they carry some, reactive molecules like superoxide ions, hydroxyl ions, hydrogen peroxides. Okay, so these peptides have the capacity to bind or to uh, remove, or you can say they neutralize these kind of oxidative reactive molecules, reactive oxygen species, and your cell will get uh, you know they get prevented from these kind of attacks by these reactive oxygen species. And it prevent also some cardiovascular disorders, neurodegenerative conditions, even in cancer also. So these are the antioxidant peptides derived from the different plant sources like black soybean, quinoa, rice bran protein, soya protein isolate, wheat germ proteins, quinoa, quinoa proteins. So these are all antioxidative peptides. They are being released from the soya protein isolate or quinoa or different black soybean. So these are the different plant-based foods or plant-based proteins through which these peptides are generated, and these peptides have an antioxidant effect. So one study I have taken about the antioxidant peptides from edible plants like chickpea and African yam bean. So these two uh, cereals, they produce this kind of peptide sequence, and they showed very good uh, results on antioxidative activity like DPPH or hydroxyl free radical scavenging activity or superoxide free radical scavenging activity. You can see they are promising peptides which are showing good antioxidant activity. Then another study on antioxidant peptides obtained from tomato wastes. Tomato waste proteins fermented with Bacillus subtilis, A14H. Okay, so these Bacillus, it is it is a spore formers. So they have fermented the tomato seed milk. The tomato seeds are sun dried, ground in a blender, and defatted using the N-hexane as a solvent. And the resulting powder tomato seed is contains around 20 to 33.9% protein on dry basis. It was used as a source of carbon and nitrogen during the fermentation with this highly proteolytic strain Bacillus subtilis. And fermentation was carried out at the rate of 2% when the number of cells will be around 10 to the power 8 in 5% trichoderma selective media and kept for 24 hours at 37 degrees Celsius. And after incubations, the fermented media was heated in boiling water for 15 minutes in order to stop the enzymatic reactions. And they have found these peptides from these tomato waste proteins after fermentations. And you can see their molecular mass and DPPH means antioxidative activities. And you can see GQ, BPP, these peptides showed 97% antioxidant activity measured through the DPPH method. Then another biofunctional properties of plant-based fermented foods is the anti-diabetic peptides. So we know that diabetes is a uh, you know in India it is basically the dreaded disease nowadays, and approximately 537 million adults aged in between 20 to 79 years are living with diabetes. And the total number of people living with diabetes is projected to rise 643 million by 2030 and 783 millions by 2045. So you can see the diabetes is increasing in a very alarming rate, and we should think of that how we can prevent this disease, because this disease is mostly spread all over the country. And we know how to diagnose the diabetes. We know the blood sugar level, fasting blood sugar level. In normal cases, it will be around 70 to 99, but after 2 hours, means after eating of 2 hours, it should be less than 140 milligram per DL. But when it will be hyperglycemia or diabetes, then it will be very high, 100 to 125 for fasting, fasting blood glucose sugar level, and 70 milligram per day should be less than after the low blood sugar level. So this is the process to check the blood sugar level in our body. Now, I will discuss that how these peptides can intervene to prevent this kind of disease. So we know these different enzymes basically participate to release the glucose from the different food materials. Okay, so one enzyme is the alpha amylase, another one alpha glucosidase, and DPP4. So alpha amylase and alpha glucosidase, they basically release the glucose from different maltose and dextrins. So the peptides, they basically bind those enzymes in their active site by interacting with covalent bond through hydrogen bonding, and they don't allow, these peptides don't allow these enzymes to act on dextrin or maltose to release the glucose. So glucose level uh, will be restricted, or you can say the glucose will be released slowly. In case of DPP4 also, it basically activate the incretins, and incretins will give the signal to uh, produce or to release the insulin, and it will control the blood glucose levels. So these are the different mechanisms basically we tested in vitro conditions to check the efficacy of the peptides generated from different fermented foods. Maybe it's a plant sources or maybe it's animal sources, basically the milk sources. Then one studies on anti-diabetic effect of the bioactive peptides, okay, from the legume sources. So here they have taken three different food materials: soybean, lupin, and common bean. And after fermentation with these lactobacillus rhamnosus, uh, these peptides are released, and they can inhibit 85% of alpha amylase or by the soybean, and even here you can see the common bean can inhibit around 30% alpha glucosidase. And in case of DPP4, you can see that uh, common bean, it releases two different peptides, LAPPG and KLLLRRLQ. So these peptides can have a good impact on DPP4 inhibitions. Then anti-hypertensive means the blood pressures. The hypertension is very common nowadays because of the stress, and we know the normal blood pressure 120 by 80 mmHg. So when you have a blood pressure, means high pressure, so it will be more than that. So we have to control our blood pressure. So these peptides have the effective results in reducing the hypertension by stimulating a balance between the constriction and dilation events of the large blood vessels, especially during the vascular injury and blood clotting. And to increase the blood pressure, one enzyme that is called ACE, angiotensin converting enzymes, that enzymes basically uh, increase the blood pressure by producing S2 from S1. So the peptides basically bind this enzyme ACE so that it cannot produce the AC2. I'll show you the actual uh, mechanism how these peptides help you in uh, managing these blood pressures. So basically you see the renin is an enzyme, it is coming from the kidney, okay. So that renin basically they interact with angiotensinogens or produced from the angiotensin, and then they convert angiotensinogen to angiotensin 1. But that angiotensin 1 is inactive. But when you have a high blood pressure, then one enzyme that basically comes from the lungs, and that enzyme is called ACE, angiotensin converting enzymes, and it converts the angiotensin converting or angiotensin 1 to angiotensin 2. When it is active, they creates the vasoconstrictor. Okay, so when these angiotensin 2 will get produced, then your blood pressure will get high. Okay, so your peptides will basically bind this S enzyme so that the conversion from angiotensin 1 to angiotensin 2 will be restricted, and your blood pressure level will be normal, and you won't feel the high pressure. Okay, so this is the role of the bioactive peptides to control uh, the hypertensions. So here you can see some of the plant sources like papaya, rapeseed, tofu, and they produce this kind of peptides which have a anti-hypertensive effect. This is the one review article we have written. One of my student has written this article, published in Food Bioscience. Basically, we have targeted to produce a review paper in very comprehensive manner on plant-based fermented foods and their health implications. That paper has been published 2 months back. Okay, so these are the different. We have collected all the sources to give a very up to date information about plant-based fermented foods and their health implications. watermelon, carrot. So these are all different plant sources, foods and materials, and they are being fermented with the different bacteria, lactobacillus or bacillus, and they have antioxidant activity and antimicrobial activity. So different food materials, they have used different kinds of compatible microbes, mainly lactobacillus or spores or yeast or mold. And these peptide sequences are updated to prove that they have a different biofunctional properties. This study has been conducted in my laboratory. One of my student, MTech student, conducted, produced the anti-hyper different lactobacillus. One is Lactobacillus fermentum and another is Lactobacillus casei. And these two cultures have given promising results. And then we have also extracted the peptides from these proteins through the fermentation, and then we have done SDS, 2D, and then trypsin digestion, and after that we have done the sequencing. And after sequencing, we get these kind of peptide sequences, okay, and they have a ACE inhibitory. These are the identified sequence. We have got it from these two different lactobacillus, and you can see there uh, already available. We have matched the sequence from the already available data bank. This is the chemical mass, and they have this kind of biofunctional activities, mainly the antihypertensive. Another study done by the Plianto and McKeon, they have uh, used some plant protein derived peptides to control the hypertension. So they have taken sweet potato, pickled by the lactic fermentation, brining the curd and balanced foods in common salt, inoculated with a Lactobacillus plantarum for 28 days. Then clinical trial was conducted in a group of 40 non-smoking volunteers with metabolic syndromes, means a little bit hypertension. And treatment period was around 4 weeks. Dietary supplements was made by daily administration of 500 milligram per kg of fermented sweet potato, and these fermented sweet potato are rich with two antihypertensive peptides, ITP and GFR. So these two peptides, they have given very good results. Here you can see the ACE inhibition is very high in the first one, ITP. Okay, ITP means isolation tryptophan and phenylalanine, tyrosine and phenylalanine. So, and you can see they have also controlled the uh, blood pressure also after 8 hours. Okay. Another antimicrobial peptides are also being released from the plant materials through fermentations. The six leading pathogens like E. coli, Staphylococcus, Klebsiella, Streptococcus, sorry, Acinetobacter, and Pseudomonas. So they have chosen these six test organisms, and then they have generated the antimicrobial peptides from the plant materials and tested against these six different pathogens. Okay, but when they have tested the antimicrobial activity, they have followed some mode of actions through different models: barrel step mechanism, carpet mechanism, and hormone mechanism. So this is the barrel step mechanism. So the peptides, you can see how they are getting docked with the uh, plasma membrane or cell membranes, and how they make the pore. These are the different antimicrobial peptides. First, they are getting attached with the plasma membrane, and slowly they make a pore. You can see there is a pore, so that they make a pore inside the plasma membrane, and all the internal metabolites will come out from the cells. So this is for wormhole mechanism. Also, they make a pore, and the pore is very bigger one, so that all the metabolites will come out. Then carpet mechanism. So carpet mechanism, here you can see the entire phospholipid bilayer membranes are being distorted. Okay, so easily all the internal different methods or models, or you can see soft, and they shown that how these peptides can kill the microbes. So these are some fermented foods: kimchi, then Chinese fermented radish, Chinese fermented cabbage, and they are fermented with different lactobacillus cultures. And from this fermentation, these peptide sequences are generated, and they have a very good antimicrobial effect or antibacterial effect against Bacillus cereus, Enterococcus faecalis, Vibrio allergiolyticus, and even E. coli. Then anti-cancer. Cancer is the leading cause of death worldwide. around 90 million deaths up to in 2020. Cancer is the rapid creation of abnormal cells that grow uncontrollably. We know that they say healthy cells, they follow apoptosis method, but cancer cells never follow the apoptosis. So they basically disturb our healthy cells, and slowly all the healthy cells convert to the cancer cells, and it's a very dreaded disease to the human beings. So possible side effects of radiation: when they have a cancer, then give the radiation therapy to, this is called chemotherapy, to get it recover from the cancerous disease. So but there is some side effects like diarrhea, hair loss, mouth problem, nausea. Okay, so these peptides can help in preventing these kind of cancerous cells to make them back to the healthy cells. Okay, so you can see these peptides, BP means bioactive peptides, they get internalized inside the cell, and then they disrupt the membrane, they basically kill the cancerous cells. Some of the work's already being done on cancerous cells. So anti-tumor activities and apoptosis regulatory mechanism of fermented barley extract has been done on a cell lines, human cell lines, HT29 in nude mice. The main composition of the raw barley, they have a moisture, protein is around 11.21%. Okay, on dry matter basis, they have fat and ash, and it is fermented with the lactobacillus plantarum. And these cell lines has been used H29s. Okay, so mice were treated for 30 days with the LFB. LFB means barley fermented with lactobacillus plantarum. Okay, this has been given up to 7 days and treated with a 5-fluorouracil by intraperitoneal injections. And you can see the development of tumor or cancer cells compared to the control. And these two sequence, LLPSY and RQSHFNNQP. Okay, so you can see that those which are giving these two products, 1 gram per kg per day or 2 gram per kg per day, you see the size of the these tumor cells is reduced compared to the control, or compared to the group which are being given 5 fluorouracils, which are as a carcinogen. So you can see these 1 gram and 2 gram fermented barley, which contains two different peptides, they are basically helping to control the cancer cells growth. So even people also worked on anti-cancer peptides from plant source like kimchi and fermented cucumber. They have fermented with two different lactobacillus cultures like Lactobacillus plantarum and Pediococcus pentosocius, and it releases two different peptides which have the effect against colon cancers, and also they suppress the cell proliferation, mainly the cancerous cells. Then use of plant-based peptides in functional food formulations. So around 120 peptides with antioxidant capacity from soybean-based infant formula. The infant formula based on soybean antioxidant peptides were interesting alternative to cow's milk infant formula. So these are the examination of bioactive peptides from plant sources through in vivo studies. People have taken some preclinical or clinical studies on rats or different hypertensive rats, or even into reduce the oxidative stress. So you can see they have given the plant-based foods, and these foods are rich with different peptides, and they have given for different days or weeks for particular treatment, and they have reduced the ROS, and ROS means reactive oxygen species, or also reduce the oxidative stress and even antioxidant activity. So these are the animal trial people have taken to prove that these peptides being released from the fermented foods, and they can control different kinds of metabolic syndromes. Stock here, consuming fermented plants that are rich with bioactive peptides have the potential to improve both human health and environmental sustainability. However, more studies are required to determine the exact mechanism and recommended dosage for and long-term impacts of the bioactive peptides present in these products. Further, functional fermented plant-based products rich in bioactives may be developed for the management of different lifestyle disease. So thank you all for listening patiently, and I am very much thankful again to the MBSI for giving me this opportunity to deliver the talk in these sessions. Thank you all.
Thank you, sir. Any questions or queries that you might have are welcome now. Any questions from the participants? No questions, anyone?
Hello.
Yes,
Uh, I have a question.
please ask.
So, as from a source PPT is represents or has considered lots of legumes like lentil, pigeon pea, soybeans, and some pea beans also. So there is a comparative analysis of the without the fermentation and after the post fermentation. There is a in the production of EMP or synthetic peptides.
You are asking about the after fermentation, so how these peptides are released?
No, sir. Actually, it has been promisely finding in the previous research that AMPs are promising found in legumes like gamma thionin, beta thionin, and defensin also. So,
Mm-hmm.
is the production of them is increased after the fermentation? Is it?
Yes, after fermentation. It is the AMP, means you are asking about the antimicrobial peptides, right?
Yes, sir. Yes, sir.
Hello. So it is produced
Yes,
from
sir.
pardon?
I am saying that initially, without the fermentation, the peptide has been found in the
Why
seeds
breaking?
and
Not seeds. We are talking about the after fermentations. Seeds, it is basically the intact proteins are there, but when proteolysis through the different microbes, maybe even some molds are also being used to produce those peptides, and these peptides are being tested that they can give different kinds of biofunctional properties. Then they sequence, and after getting that sequence, they check on different softwares are there. There is this database called Biopep, where you can see or match with your own sequence with the reference sequence that these peptides have antimicrobial activity, and then you can synthesize these peptides, which you got it from your own food, to basically validate the efficacy of those peptides against these different pathogens or some spoilage causing organisms to prove the antimicrobial effect. Am I
Any more
audible?
questions? Yes, sir. Yes. Any more questions from the participants?
Yes, please. I have one question, sir.
Mm-hmm.
Myself, Dr. Prabal Singh Chauhan, sir. I am just near in Vadodara, near about you.
Yes.
Yeah. Sir, actually, I just want to ask one question that have you ever seen the effect of these bioactive peptides on the fatty liver?
No, I did not check. Basically, I
Hmm.
am working on milk peptides. Basically, I am in a
Okay.
dairy biology department here
Mm-hmm.
at okay.
Mm-hmm. Mm-hmm.
So we are basically working on hypertension, then diabetes, means anti-diabetic effect, and antimicrobial also. But but what you are asking about is the NFLD. Now
Yeah.
we are thinking to explore against the NFLD on liver cells. Okay, because we are getting good results on anti-inflammation also.
Okay.
On raw macrophage cell lines, we have done
Yeah.
these peptides are very good effect against the anti-inflammations. Those inflammatory markers are there, are like IL-6, IL-10, IL-1 beta. Okay, those
Okay.
inflammatory
So
markers are there. And
yeah, yeah.
it's
So this
reducing.
shows the these peptides also showing the anti-inflammatory activity.
Yes, yes.
And I think it has been proved in the animal models.
No, we did not check on the animal model. We have
Yeah.
checked in the cell line model, raw
Okay.
macrophage 4.7, raw macrophage cell line
Okay.
on
Okay.
anti-inflammatory activity.
Thank you, sir.
Thank you. I think
So
Prakash Halami
Dr.
sir has raised a
Yes,
Halami sir, good
yes, sir,
evening.
yes.
Good evening, good morning.
Oh,
Can
you
you
are
hear me,
outside
sir?
the country.
हाँ क्या करें कुछ पेट पानी के लिए see you did not join for a round table discussion and then I have to only answer all the questions.
Actually, sir, I was in Shillong on the time, and there is a severe rain on the time, and there is a very big problem on network. I was in
No,
Nehru.
because here I have the limitation of
I
the time
informed before, sir.
availability.
No, sir.
Yes, yes.
I informed the organizer. At least give me another week to reach the Anand. Then we'll definitely. Then they give
Exactly.
this kind of this assignment, and I said yes, I will.
Not issue. No, what I was trying to ask was, of course, first initial part I missed. But how we actually look the consistency in the product? Because if you are looking for a natural fermentation, if it is not a defined starter
Yes,
culture, you look for a natural
yes, yes,
fermentation, so you don't get the consistency, right? Another thing is with reference to the safety aspect when
yes,
we
yes.
look for the natural fermentation, and then third, of course, is the regulatory aspect, which now FSSI or any regulatory agency is looking that how the end product will be consistent. Because now here actually I'm consuming suppose the kefir, which they just add few lactic cultures bifidobacteria, but I don't think kefir is really what it is naturally fermented, right?
Yes, it's
So
true,
you
sir.
use the defined culture to get the finished product of uniform consistency, but then you will not have what the actually the product supposed to be, right?
Yes,
So in that
it,
that aspect, what
true,
is
sir.
your opinion?
Your opinion or your suggestion, I will say it's very valid because
Mm-hmm.
uh, as we talk about probiotics also, when we'll claim for a particular strain, maybe we
Yes,
don't know exactly what happens when it colonizes in the gut. Okay, so
yes.
even in the peptides also, but the thing
Mm-hmm.
is that if you can purify these peptides, because you see that we are talking about the peptides, not exactly the food here. Food is a function. When it is a functional food, then obviously there is a question about the concentration of the peptides, then peptide structural things, and the you said that the the stability of the peptides after the
Yes,
fermentations
yes,
and
yes,
bioacceptability. But
yes.
the best way to deliver the peptide is that you purify it and then you sequence
Uh,
those peptides, and you think that which peptides are more the potent. Through different softwares we can analyze this, and then
mm.
what you have to do, you have to synthesize these peptides. As we are now working with IIT Ropar, they are
Okay,
synthesizing
okay.
some of my peptides. Okay,
Mm-hmm.
and that peptides, then I will quantify how much these particular peptides are in my product. Okay, because you know about the calpis. Calpis is a basically a product,
Yes,
fermented milk
yes,
product. It is
yes,
called functional food also,
yes.
and it is basically developed in Japan. Okay,
Mm.
and they have fermented with the combination of lactobacillus and yeast Saccharomyces cerevisiae. And there are two peptides on this product, IPP and VPP, isolation
Yes,
proline proline and valine proline proline. And they have quantified it, that how much present of these two peptides in that particular
mm.
product. And they have done clinical trials on this particular drinks. Basically, it's a drink, my fermented milk drink. and it has a significant impact on control of the hypertension. And these two peptides are very
Okay.
much, potent to manage these hypertension. So we have to think of in that level that how your product, but when you say it's a natural fermented foods, then we have to be very careful because we are using all defined systems. But when you
Exactly.
will make a functional food with a rich peptides, that can be possible. But again, we have to go because the regulatory aspects. And most important part is the dosage, that how much we should need to control this kind of hypertension. But from my, you can say it's a really, I will not say that it is a clinical trial, but when I am having doctors say that you may have some kind of, pre-hypertensive like things. So I started to take fermented milk. Basically, it is called lassi like buttermilk. And that buttermilk is fermented with 3 bacteria. It is sold by Amul at Anand. Okay. They have a 3 bacteria. One is Lactobacillus acidophilus, then Streptococcus thermophilus, and Bifidobacterium bifida. Okay. These 3 bacteria, it is fermented with buttermilk. And when you will keep that buttermilk in your refrigerated conditions for long time is more than 10-15 days. Okay. So these bacteria grow and they produce lot of acetic acids also, because the
Yes.
taste will get changed, because and it has a very good impact on your health if you have some kind of symptoms of hypertension. But I am not saying it is validated, but I am feeling better when I keep this buttermilk for long time. Maybe their prescribed shelf life is 10 days, but I am keeping for 15 days. Okay. And then when I started to take at least 100 ml per day, it has a good impact on health. But
yeah, I think we
even
can
you can say it's the impact of
discuss.
bioactive
I think this
peptides or
aspect.
short chain fatty acid. There is
Yes, we can discuss this
huh?
aspect subsequently later.
Yes,
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 yes.
sir.
Especially that
Because it is why actually, sir. I thought I actually used the concept of sampan.
okay.
The sampan is very costly because they are fermenting for 1 year. Okay.
Okay.
So
Okay.
I also proposed these things to Amul also, but what they suggested that the customers love the taste, because when you keep for more time, because you know that you are working, sir, with the bifidobacter, they produce good amount of short chain fatty acids, mainly
Exactly.
the butyrate. So a little
Yeah.
bit and acetic acids. A little bit taste is being changed
Yes.
because of and these are heterofermentative bacteria, so they produce some amount of carbon dioxide and hydrogen also. Okay, but it has a very good health impact. And I used to take for around 6 months, and when I again checked, they said you don't require even in medicine or anything like that. It's a very good effect. I know doctors said, and they said you don't need to take anything right now.
Very
So
useful. Very useful.
yeah, but
Soya
depends on
fermentation.
yeah. What about?
Yeah, soya milk. Suppose if we use for fermentation,
Yes, sir.
only bifidobacteria are enough, or we should have the bacillus or lactobacillus?
I think
Because
that
protein.
one bacillus like acidophilus or helveticus, this combination will have a more impact
Exactly.
on releasing these metabolites.
Yes. Yes.
Because bifidobacteria is an excellent bacteria, no doubt about it. In case of probiotics, I'm telling you.
Yes.
But when you
Yes.
talk about the proteolysis, because
Yes.
peptides and lactose and
Very,
other things,
very
yeah.
limited proteolysis actually.
Yes, very limited in bifidobacteria. So
Yes.
to enhance this kind of activities, you have to have some other combination. It will have good impact, because I
Yes.
experienced this one.
We discuss this part later on.
Yes,
Yes.
yes.
Thank you.
Now,
Thank
now I request Imani ma'am. We can conclude now. There was a discussion. There was a good presentation. So,
Yes, sir.
Thank you, sir.
Imani,
Sir, thank you so much for introducing us to multiple new trends with respect to fermented plant-based foods. We are very grateful to be a part of this session. On behalf of Microbiologist Society India, I would like to extend my heartfelt gratitude to Dr. Shubrata Hathi sir, Assistant Professor, Department of Dairy Microbiology, SMC College of Dairy Sciences, Kamdenu University, Anand, Gujarat, India. Sincere thanks to Dr. A.M. Deshmukh sir, All India President, India, for organizing this event. Finally, a huge thanks to all the participants. Thank you so much, everyone.
Thank you. Thank you very
Thank you.
much.
Thank you all. Thank you, Deshmukh sir.
Thank you. Thank
Thank you,
you.
Halami sir. Good night.
Yes, for excellent presentation.
Thank you. Thank you, sir.
Tomorrow it will be circulated.
Yes.
Okay.